Hello and welcome students. Today we are going to discuss the remaining part of topic component and tools of IPM. In that we discuss the last part, that is chemical control. In that we discuss here why we use the chemical control or when we use the chemical control. When all the methods like cultural methods, physical methods, biological methods, mechanical methods, when all that methods are not efficient for the management of that pest or not effective for the control of that pest or disease, at that time we have to use chemical control to keep the pest population below the economic injury level to avoid the further loss. So we discuss here what are the methods of chemical control to the seeds and seedling. So in that first we discuss about dry seed treatment, then wet seed treatment and slurry treatment. So it is a very simple method. In case of dry seed treatment, we have to use a powder formulation of fungicides or insecticides. Take a required quantity of chemical, it may be a fungicide or insecticide, and take a required quantity of seeds which is to be treated. For example, we have to use bivestin at a rate of 2 gram per liter of water. So simply we have to take a required quantity of chemicals and required quantity of seeds. Then both are mixed with each other in a one pot and add a water in a such a way that chemical or that dry powder of that pesticides or fungicide is completely covered the outer coat of the seeds, and after that uniform coating of fungicide or insecticide over the seeds. Such as seeds are dried for certain period, and after that such as seeds are used for the sowing. So this is about the dry seed treatment. Next one, wet seed treatment. That wet seed treatment is used for the control of different kinds of soil borne as well as seed borne diseases. So that wet treatment is used for the rice, wheat, for the roots, or sometimes planting materials like rhizome or set or suckers. Then let us see one by one. Here for wet seed treatment, if you take the example of out of that 4 or 5, if you take the example of wheat, so in that we have to take the appropriate quantity of carboxyn. As we know that the rate of carboxyn is 0.2%. That is 2 gram of carboxyn powder we have to mix with 1 liter of water. In that water we add the required quantity of seeds. Keep that seed as it is in carboxyn solution for a period of 10 minutes. And after that 10 minutes we have to remove the seeds dried in shade and then directly used for the sowing. So such a carboxyn, that treatment we have to use to avoid the infection of loose smut of wheat, which is caused by the fungus Ustilago triticide. Next, again we take the example of rhizome dip. So simply the fungicidal solution, that is emission solution, is prepared at a rate of 0.1%. That is 1 gram emission solution is mixed with 1 liter of water. In that the rhizome of banana is dipped for a period of 5 minutes. After that rhizome is used. Similarly, the sets of sugarcane and the suckers of pineapple is dipped in 0.1% emission solution for 8 to 10 minutes. Then after that treatment, that sets or sucker is used for the planting. So in that way we have to use the wet seed treatment to avoid the seed borne or seedling borne diseases. Next we discuss about the slurry treatment. So as we know that slurry treatment, just like a dry seed treatment, we have to take required quantity of chemical, it may be fungicide or insecticide, then take required quantity of seeds, both are mixed with each other in a plastic bucket or in a pot, then add a water in a such a way that slurry is formed. So when slurry is prepared, so such a slurry that are directly placed on seeds, or seeds and such a slurry that are mixed in a such a way that uniform covering of or uniform coating of such a chemical is formed on the outer surface of seeds. Then such a seeds are used for the drying for 5 to 10 minutes, and after that 5 to 10 minutes or 15 minutes depending on the environment, that seeds are used for the slurry sowing. So this is about slurry treatment. Then last one is acid delineating in cotton. So why we do the acid, why we use the acid for in cotton? Because you know that cotton having a fuzz, that is fuzz is a genetic characteristics of seed of cotton. Fuzz is nothing but a hairy or whitish growth is observed on outer surface of seed of cotton. When such a fuzzy seeds are used for the sowing, so germination of such a seed is not takes place properly. So to avoid that, we have to use acid delineating in cotton for better germination and to break down the hard seed coat of cotton. For that, we have to use 100 ml of H2SO4 solution, that is concentrated H2SO4 solution. 1000 ml of water we have to taken into bucket, and required quantity of seeds. So simply in 1000 ml of water, we have to add 100 ml of seeds, 100 ml of concentrated H2SO4. Then with the help of wooden material, we have to thoroughly mix with each other so that seeds and that H2SO4 solution get mixed in that water. Then after that mixing, we have to keep as it is such a seeds in a bucket for 2 to 3 minutes. Within 2 to 3 minutes, whatever the floating material which is present, that contains light seeds or broken seeds or infected seed that are floated over the water or H2SO4 suspension solution, that suspension is discarded. Then remove the excess solution and take the healthy seeds which is present at the bottom or accumulated at the bottom of solution or bottom of bucket. Then such seeds are directly transferred into 2 to 3 plates which containing water for washing. That is, wash such seeds 2 to 3 times to remove the traces of concentrated H2SO4 solution. After that, we have to dry the seeds, and after drying such seeds, fuzz of seed is completely removed due to acidic treatment, and such a seed are directly used for the sowing. So in that way we have to do the acid delineating in cotton. So certain precautions we have to taken while acid delineating in cotton, that is use plastic bucket, because we have to use concentrated H2SO4 solution. So if you take the metal bucket, so it causes corrosion or it causes a reaction with that metals. Then for stirring we have to use wooden material instead of your hand. And third one, you have to wash 2 to 3 times to remove the traces of wash the seeds of cotton 2 to 3 times to remove the traces of H2SO4 solution. So if you take such a precautions, so that treatment is possible. So this is about the chemical seed treatment. So if you have any difficulty in that components or tools of IPM, so you can ask on my personal WhatsApp number. That topic is very important for the exam point of view or for objective point of view. Thank you.